What we did in the study was to kind of piggyback on a study that our colleague Ben Levine did at his center, where they collaborated with NASA to do a 5-week bed rest study. So they had people in a bed resting with or without exercise. For those that didn't get to exercise, pretty hardcore muscle deconditioning, which is supposed to kind of mimic spaceflight. So they did muscle biopsies before and after, and what we did was RNA sequencing on the muscle biopsies, and we looked for changes in gene expression in the muscle after 5 weeks of deconditioning. So then what we did was to take that data and use this ligand receptor interactome that we've developed and see which of the things that were increased in the deconditioned muscle might act on nociceptors. And what we found was pretty interesting. We found basically that the deconditioned muscle upregulates a bunch of inflammatory cytokines and other types of inflammatory mediators, many of which could potentially act on nociceptors to induce sensitization. Many of the things that we found are not things that people typically are studying. We wouldn't have known that they could act on nociceptors unless we had this really nice data set from our organ donor work on what receptors human nociceptors express. This study lays a groundwork to do some really neat and interesting things on understanding how muscle deconditioning may lead to widespread sensitization of nociceptors and could help promote pain chronification.